Hi, everyone. My name is Jennifer Nosker, and we are pleased to meet you and to have our No Neuropsychology Didactic series today. Our series is planned by the No Neuropsychology Committee, as well as the No Neuroanatomy Committee. And we would also like to let you know that the series was created by trainees and early career neuropsychologists to provide high quality didactic opportunities. And we would like to go ahead and thank our sponsors for their contribution to our series. Now, before we start, we want to make sure that everyone knows about our YouTube channel. All right. And with our YouTube channel, every No Neuropsychology and No Neuroanatomy lecture is available for your viewing pleasure. So be sure to go check it out, subscribe, and like our lectures. And here's our disclaimers for the series. Now, this training is not meant to replace formal education in neuropsychology, and the views of the speakers are their own. Now, questions can be submitted via the Q&A box on the lower left of your screen, and a recording of today's lectures will be provided on our website and YouTube channel later this week. Now, it is my pleasure to introduce Dr. Robin Lester for today's lecture on white matter pathways. Dr. Lester is a cellular neurophysiologist interested in the role of ion channels, receptors, and synapses in learning and memory and addiction. In the past, his lab has utilized physiological techniques, patch clamp, CA2 plus imaging to investigate the physiological properties and regulation of various subtypes of CNS nicotinic and acetylcholine receptors, slow cholinergic nicotinic receptor-mediated synaptic transmission in the limbic system, and physiological adaptations in the hippocampus following withdrawal from chronic nicotine. Over the last 10 years, he has transitioned into a full-time teaching position focused on graduate, undergraduate, and professional education. Dr. Lester has developed courses on the biophysics of ion channels, synaptic transmission, the history of neuroscience, human neuroanatomy, and addiction, with a philosophy that encourages student engagement and the use of online resources, including TED Talks, science podcasts, and lay scientific articles. And with that, I will go ahead and hand things over to you, Dr. Lester. Thank you so much for speaking with us and presenting today.
Well, thank you for the invitation and the opportunity to discuss this topic. I will share my screen.
That looks great.
Okay, so as Jennifer said, originally a physiologist, and I've become more recently interested in neuroanatomy, human neuroanatomy in particular. And I have a bit of a thing for the history of neuroscience. So we might take a little bit of a convoluted journey towards white matter pathways today, but we'll get there. So this is an outline of my talk. I'd just like to start off talking a little bit about how the nervous system is organized overall, and then take you through how we got to the need for white matter pathways. And I'll talk specifically about a few of those pathways and some of the things that may go wrong with them. Finishing off with maybe a special case of myelinated pathways, and then just a few words on where we might go in the future. Okay, so let's start off with a question. So, what is the longest cell in the body? Well, we're talking about neuroscience here, so pretty much everybody's going to guess that it's a neuron, and in particular, the axonal part of the neuron. But we might have to look outside humans if we're going to actually find which animal has the longest cell. And a bunch of scholars have looked into other animals, right? Obviously giraffes, and larger animals, whales, and even going back to dinosaurs. And so it's been hypothesized that in some of these dinosaurs that you might have an axon which stretches from the tail all the way to the brainstem. And this could be as long as 30 meters in some of these creatures, which, based on a signal speed of about 1 meter per second would potentially be problematic. So in an uncoated axon, right, the signal could take from the tail, if the animal was touched on its tail, about 30 seconds to get to the brain and to be interpreted. So we'll see how that problem may go away if we make use of other glial cells in the body. The equivalent in humans is the sciatic nerve, which is going to be greater than about 1 meter, which obviously, again, would still be a bit of a problem if we rely on those sensory signals that are going to be taking a second or so to reach the brain. Let's go back just for a second, a little bit further, maybe to around 3.5 billion years ago, when the only organisms around were single-cell creatures. But I want to introduce a topic which really spans all the way to humans, and that is the idea of sensory-motor integration. So even in these single-celled organisms, right, they were able to sense, for example, nutrients that they would be attracted towards and could move towards, or away from toxins, be repelled and move away from those again. So contained within a single cell was all the necessary equipment for sensory motor processing. As organisms became bigger, they started presumably to separate sensory motor functions into different cell types. And that wasn't necessarily a problem. If they were close together, they could communicate by diffusion of chemicals. But as those organisms grew, those cells would have to become further apart. And it's hypothesized that that's what caused the need for neurons, and in particular, long processes to grow so these different cells could communicate efficiently. This started to happen about 500 million years ago, and the first organisms, or amongst the first organisms to have neurons, were creatures like jellyfish. And jellyfish communicated by having networks of relatively short neurons that were able to do a lot of processing. If we move quickly to humans, right, it's not that much different. So humans, we have a central nervous system, which comprises the brain, and sometimes we forget about the spinal cord. And then a peripheral nervous system, which is really about bringing in sensory information from the periphery and communicating back out motor signals to our muscles, for example, to cause us to move. So yes, there's been a lot of evolution and probably a lot more integrative processing in our CNS, but the basic principles still hold up. Over the last couple of thousand years, various scholars wanted to investigate the nervous system in more detail. And actually, what they first started looking at was really the same story, the story of sensory and motor processing. And in particular focused on reflexes. And Descartes was the first philosopher scientist to really start proposing a mechanism to account for reflexes. Most of you are probably familiar with this Descartes figure, which discusses the withdrawal of the foot from a noxious or painful stimulus. And we already knew that nerves went to and from the spinal cord and brain. We didn't know much about those nerves. In fact, they were proposed ever since Greek and Roman times to be hollow tubes full of what they called animal spirits or pneuma. So nothing like we know today. And Descartes proposed there was some sort of thread-like mechanism that existed within these tubes and would open a gate up in the ventricular system, and that would let animal spirits flow back out and cause withdrawal of the foot by inflating the muscles. So of course, we know at this point that that mechanism is likely to be quite wrong, but it provided the groundwork for future studies. So jumping forward to not so long ago, really, right? Just a couple of hundred years ago, we saw the merging of scientific disciplines, in particular, the fascination with natural electricity. So scholars like Benjamin Franklin, right? Everybody knows the famous kite and key experiment, but he was able to show that he could capture electricity and store it in jars. And once electricity was able to be stored or generated, it could then be used to cause other processes to function. And it took a few other people to start showing that electricity could be shown to move along threads or wires, and different substances had different conductive properties. So it would clearly move better along wires than it would along threads. But this religious scholar, Stephen Hales, was the first person, as far as we can tell, to propose that electricity was used to conduct signals along the nerves. And then Luigi Galvani, and often forgotten was his wife, who were both scientists, that first demonstrated that they could get frogs' legs to move by stimulating the nerves in the spinal cord. And of course, the ideas potentially behind Mary Shelley's novel, Frankenstein's Monster, was really provided by Galvani's nephew, who decided it would be a good idea to reanimate recently executed humans. And this was, something in Europe that was quite popular and, would spread. So clearly at the same time, Mary Shelley would be aware of these experiments. Although if you go back to her original book, she never said that Frankenstein's monster was reawakened by lightning strikes, for example. I think that came in the movies later on. Shortly after that, it was Helmholtz who actually measured the speed of conduction. And that's when it was realized that bioelectricity was a lot different than physical electricity, because it moved at a fraction of the speed of light. And then, just at the turn of the 20th century, Bernstein was able to demonstrate that this bioelectricity involved ions, sodium and potassium ions, moving in and out of cells. And that whole process was time consuming and energy dependent, and was the reason why nerves can't conduct like we think of electricity moving down a wire. But soon after that, or around the same time when electricity was proposed to be the conducting substance or mechanism, there was a lot of concern, principally from Haller, who was worried that electricity would just leak out of cells and go everywhere. But if we step back to Leeuwenhoek's microscopic studies, he'd already started to observe that there seemed to be fat coating the nerves. And in fact, even Galvani had seemed to find that there was an oily substance or something else that was coating nerves. And perhaps that would contain electricity to within nerves themselves, although at that time, nobody had discovered individual axons. But at least it provided some reassurance that, that electricity could be contained and therefore directed. And then it was the mid-19th century when Virchow first described the coating myelin. So myelin, you know, the coating is not needed necessarily just for insulation. What it also does is affect the speed of conduction. And I'm not going to go into all the details here, but basically to improve the speed of signal transfer in neurons or axons specifically, you can do a couple of things. You can increase the axon diameter. So just like water will flow better through a wider diameter pipe than a small diameter pipe, so will electricity flow through a larger axon. And human axons range from about 1 to 20 microns. If you jump to squid, and squids don't have myelin, but they've evolved a very large diameter axon called the giant axon, which is about 1,000 times bigger, 100 to 1,000 times bigger than human axons. And so they can conduct pretty fast when they need to. So the myelin insulation, which you can see here, coating the axons, is a very good way of increasing the speed of conduction. Because as I mentioned, conduction depends on ion channels opening and closing. And if you can restrict them to just a very small part of the axon, you only need to regenerate the signal at, you know, pretty large distances along the axon, and this can speed up transmission significantly. So you can go from about 1 meter per second to over 100 meters per second. So this gets rid of the problem in dinosaurs, right? That signal will get from the tail to the brain pretty quickly. So in humans, we have 2 main types of myelin cells, the Schwann cells and the oligodendrocytes. The Schwann cells are in the peripheral nervous system, and there's a 1-to-1 wrapping around of the axon. And you can wrap around, you know, 50 to 100 layers of myelin. So these are very well insulated wires. The oligodendrocytes are found in the CNS, and presumably because there's a lot less space in the brain, right? Because it's contained by the skull. There's not a 1-to-1 relationship between oligodendrocytes and axons, and they can coat a lot more axons, which means if you lose oligodendrocytes, you're going to cause quite a disruption to signaling in the CNS. So that takes us to gray and white matter. And these are 2 images, one a coronal section of the brain, and one a transverse section through the spinal cord. Gray matter, right, as you can see here in the brain, contains cell bodies, dendrites, synapses, and a lot of glial cells like astrocytes. But it's these white matter tracts, right, that are bundles of axons. And they appear white because that's how myelin appears naturally. So these are all white matter tracts. And roughly speaking, there's about 50% gray matter and 50% white matter in the CNS. Just to confuse everybody, I've picked a picture here where the myelin is stained dark. So in the brain, right, you can see the gray matter is on the surface of the cortex and the white matter tracts are internal. Whereas in the spinal cord, it's roughly the opposite way around. The tracts, these long pathways are in the outer layers, and it's the inner layers that have the cell bodies. So just as a quick break and from all of that, somebody told me, or I read this somewhere, and I failed so far to find out where that's from, that you couldn't have either very large diameter axons or myelinate every axon in the CNS because your brain would be as big as the moon. I'm sure that's not true, but you kind of get the picture, right? So we have to restrict these wide diameter axons that are heavily myelinated to certain pathways in the brain. And it seems like a lot of those pathways are covering long distances. So the types of connections can be categorized as either association tracts. So within one hemisphere of the brain, you have connections from one part of the cortex to another part, and we call those association. Then there are commissural tracts, of which the most famous is the corpus callosum, which connects the two hemispheres, and then projection tracts, which basically connect the cortex with deeper underlying structures and even the brainstem and spinal cord. So this is just to give you a quick appreciation of the different white matter tracts in the brain. We're not going to talk about all of these today. Some of them are found in limbic system structures. We're going to talk initially about the major sensory motor tracts, and then talk about the tracts that are important for language and communication, and then talk about briefly the corpus callosum, the commissural tracts. So the general rule here is that, at least in the mammalian brain, in particular in humans, is that it doesn't matter what type of information you are. You can be motor, sensory, limbic, cognitive information. If you want to get to and from the cortex, you have to go through a pathway called the internal capsule, which is shown here in axial section as a V. If you take a slice down here in this orientation, sagittally, and look at the pathway side on, you can see that this pathway radiates out from this central spot, right, coming from down here, and all the way to every part of the cortical surface. And it's carrying all types of different information, largely upwards, but not exclusively. You can see that nicely here, all coming into the internal capsule and down into the brainstem and spinal cord. So we're going to focus on that pathway right now. And I've just broken it down into the major components. And you can see how much of both the brain and the spinal cord this covers. So you can see, for example, the motor pathway originating in the motor strip of the motor cortex over here. It travels down and it goes through the internal capsule and then through the brainstem here, where it crosses over to the other side in the pyramidal decussation and down through its specific part of the spinal cord, where it eventually synapses and connects to muscles. So you can see this is a very long nerve that goes from the motor cortex all the way down into the spinal cord. And pretty much connects, right, every muscle with some part of the motor cortex. The same is true for ascending sensory pathways, where sensation comes in and then finds its part of the spinal cord, goes up and, or in some instances, crosses in the spinal cord and then goes up into somatosensory cortex. Major sensory pathways are generally divided into two types. One taking touch and proprioceptive information, and one taking pain and temperature. And they have slightly different axonal and myelination properties, because you probably know that a lot of pain signals can take a little bit longer than touch and proprioception signals. And it may not be immediately obvious why proprioception has to be transmitted quickly. So those axons are very wide diameter and heavily myelinated and take a big chunk of the spinal cord. And it's because, right, we can't regulate our movements very well if we don't know where our body is in space. So this sensory motor integration, right, requires heavily myelinated white matter tracts that take signals very quickly all the way back to the somatosensory cortex, and then back out to the motor cortex and back down to the muscles. Just one general point is that you notice that all of these pathways cross over to the other side of the CNS at some point, so that representation of the body is contralateral in the brain. Okay, so quickly on to association pathways, and a pathway of choice is the arcuate fasciculus. The arcuate fasciculus is part of this superior longitudinal fasciculus, which carries a lot of fibers of different sources and destinations to and from the anterior part of the brain to the more posterior parts. Everybody is probably pretty familiar with Broca's area, right? One of the first areas in the brain that turned the focus of a lot of physiologists and anatomists to the idea that functions were localized to specific parts of the brain itself, right? And so language output or speech, right, was localized to Broca's area. Whereas language input, right, and comprehension was localized to Wernicke's area. But this pathway, the arcuate fasciculus, was important to connect the two. So again, it's sort of a sensory motor pathway, right? Sensory, auditory information and processing coming in, and then to have a conversation, right, we'd have to send that information back out to form speech. Damage to this pathway, right, or any of the language areas, as you probably know, when somebody has a stroke on the left side of their brain, can be devastating. But you can have selective damage to the pathway itself. And what happens then is a person can understand language because Wernicke's is unaffected. So if they can't tell you they understand language, right? Because the language pathway cannot get back to the speech output. But if you ask them to perform an act, such as point at something, they'll be able to do that. But even though Broca's area is unaffected, they'll have nice fluent speech, but it won't make any sense because they're not getting information from the comprehension parts of the brain. So they'll talk fine, but it'll be kind of gibberish. So let's finally move on to the commissural tracts, and I've picked the corpus callosum, which you can see dissected out nicely here, right? So you can see there are massive connections from one half of the brain to the other, which actually is still a puzzle in some ways to this day, because even back, almost 100 years ago, there was neurosurgery in the early part of the 20th century that involved cutting the corpus callosum to prevent seizures spreading from one side of the brain to the other. And there was a lot of discussion, but a lot of these patients didn't seem to have much functional loss. But if we go back to, if we go a little bit further and we start looking at that in more detail, as Roger Sperry and Gazzaniga did in the 60s and 70s, they found there were actual problematic deficits, right, as you might expect. And I'm just going to show you the most simple one here, right? If you have a split brain where there's no cutting of the, where there is cutting of the corpus callosum, then information theoretically can't get from one side to the other. If you present an apple, for example, in the right visual field, and that information goes back to the left occipital visual cortex, the person can tell you, right, that they saw an apple because they're connected through the association pathways to the speech centers. But if you present something in the left visual field, like a pencil, it won't, it'll get all the way back to the visual areas on the right side, but the person will not be able to tell you what they saw, even though they will be able to demonstrate what they saw if you, for example, ask them to select an object. And I just historically here, right? This idea of dual minds has been around for a long time, right? Probably made most famous by the book Dr. Jekyll and Mr. Hyde by Stevenson. But this idea, right, continues to the current day, right? Whereby the right hemisphere does certain things and the left hemisphere does other things, right? Traditionally, it was viewed that the left hemisphere is civilized and intellectual and the right hemisphere is uncivilized, right? But, you know, we still have that hangover today, right? When we're asked to take those surveys, are you right or left brained? Some of you might be interested to know that other scientists decided to say that the right side was the female brain and the left side was the male brain. And you could see where that might take us, right? If we're still hooked up with the whole Jekyll and Hyde idea. Okay, so I just want to finish off with a case. So I've tried to show you that, you know, it's important, right, to have these heavily myelinated pathways to cover long distances because you want communication to be relatively fast, right? But there are also short pathways in the brain that need to be fast or almost instantaneous. And so we don't just restrict myelinated white matter tracts to these long distance, to cover these long distances. So this is a case where a patient had a weird problem with their eye movements. So when this patient was asked to look to the left, both eyes moved to the left. But when they were asked to look to the right, the left eye, the left eye, I always get mixed up here. So this is the right eye. So this eye, right, couldn't move past midline, but the other eye could move to the right, right? So this is the right eye, the left eye couldn't move, the right eye could move. So there was something seemingly wrong with the left eye. And it turned out that the pathway that was disrupted was something called the medial longitudinal fasciculus. And this is a pathway that's really important because it has to coordinate the movement of both eyes. In fact, produce conjugate instantaneous movements, right? Because if you don't move both your eyes together, you're going to have double vision. You're going to have blurred vision. You're not going to be able to focus an image. So this is a problem that can occur in multiple sclerosis and is often diagnostic. You can see there's a plaque here in the mid-pons that's disrupting this myelinated connection, which is not very long, but it has a very important and devastating problem. So I know you're going to have another seminar in a week maybe or so that's going to discuss networks, but some of you are probably familiar with how we look at myelinated pathways in the brain using diffusion imaging, diffusion weighted or diffusion tensor radiography. And I've picked a few images here to show you how a lot of the times we see them, right? They're color coded, as in those really beautiful pictures showing the human connectome. But you probably also know that we don't really fully understand what all those pathways do. And we're just starting to get an idea about how the brain is fully connected up to perform whatever behaviors it has to perform. So this lets us revisit an idea of localization versus holism, a debate that was prominent in the 19th century, right? Because Broca had found Broca's speech center and promoted the idea that we could localize functions to specific parts of the brain. But the whole other side of the debate was about holism. And when other scientists started to ask where memory or intellect was, they found that very specific lesions couldn't disrupt some of those cognitive functions, and led them to the idea that some higher order functions were distributed throughout the brain, and perhaps even with some redundancy. But I think we're kind of, at the moment, maybe thinking that we're somewhere in the middle, right? There are localized areas where certain functions go on. And, you know, some of these pathways are probably heavily myelinated. You can see them in cortical regions going from one part of the cortex to something close by. But maybe some of the most heavily myelinated pathways are these projection ones. So this allows, for example, right, us to have a small group of friends, right, who are in close contact with, but to be able to jump quickly maybe to a different set of friends because we're connected, we have a common friend that's connected to both of these small areas. So we have this idea of small world networks, and I'm sure Dr. Vischer is going to talk a little bit more about those in the near future. Okay, so I would like to thank you for listening to me, and maybe you can contemplate how fast we think and what sort of pathways it would use. I'd like to acknowledge most of the images I've gotten from these books. One on the history of neuroscience, a couple of neuroanatomy books, and this, one of my favorite for illustrating areas of the brain, and particularly the pathways, is this photographic guide to the human brain, which I recommend because they've dissolved and dissected out all of these white matter pathways. And you can really see how much coverage they have inside the brain. And I will end there and thank you for your attention again.
Well, thank you so much, Dr. Lester. That was just an absolutely fabulous lecture. And I have to say, I absolutely appreciate your "don't waste your potential" picture on your last slide. So we did actually have a question, and it has to do with your research. So a lot of our attendees are neuropsychology trainees, and so many of us want to know the impact of different diseases or disorders on different parts of the brain. Given your research in addiction, what does substance use or the effect substance abuse have specifically on white matter pathways and tracts?
Yeah, that's a great question. And I really don't think we have much of an answer to that at the moment. But I can rethink it in a slightly different way. there's probably not any part of the brain that's unaffected by addiction itself. It's a whole brain phenomena. It definitely influences one major tract, which goes from the brainstem to the forebrain. Everybody knows that is the reward pathway. But, right, it's also a prefrontal cortical problem with deep, heavily myelinated descending pathways. Some of those pathways we think are disrupted in addiction, don't work as well, are late to develop, and so may explain addiction in adolescence. So I think it gets back to the idea that there are many different parts of the brain that regulate motivated behaviors. And so it fits in with this idea of, a little bit with this idea of small world networks, where some aspects of motivation go on in particular parts of the brain, but they're still in communication with the rest. There was a recent study that came out where they looked at some parts of the brain, in particular an area called the insular cortex, which had been known from anecdotal cases to be kind of strangely involved in addiction when people had strokes in that region. What they found was if they were smokers, right, nicotine addicts, when they recovered, they never went near cigarettes again. So it was kind of an unpredicted area, but now it's been re-examined with radiographic imaging, and it's found that it's not just the insula, right, which could be regarded as this gray matter chunk of cortex, but it's also its connectivity to other parts of the cortex that are and deeper structures that are really important. So kind of a long answer, but
Well, it's really helpful. And again, it's almost like the more you learn in this field, the more you realize we don't know, right? When there's just so much more research that needs to be done.
yeah, that's very true.
And, again, for many of our trainees who are interested in learning more, you do have a couple of great books listed. I'm wondering if you were to recommend one specifically on white matter tracts, or even just kind of an easy, neuroanatomy is never easy. It's quite complex, but kind of a good go-to. Is there one that you would recommend for our trainees?
If you're interested in human anatomy, either of these books, both of these are sort of a bit more medical in their context, particularly this one, which is a neurologist who discusses a lot of clinical cases. It's still a great reference book, but it's big. It's about 900 pages, so I use it as a reference. This book is much easier to get through. It discusses all the major pathways in the brain, different areas and different functions. It goes all the way from cellular physiology and cytology all the way up to behavior. So I like this. And there's even, in fact, a compact version of this book that's very digestible. So I mean, but there are many other neuroscience texts, right? But if we're focusing on humans, I like these two best.
That's excellent. Thank you so much for that recommendation. I know, for me personally, I've taken neuroanatomy course, I've TA'd for neuroanatomy courses through my university, but I still feel like it's just, there's so much to neuroanatomy, both structural and functional, that I need like not a quick reference, but something that sometimes that I can refer to and maybe digest a little bit easier. So I personally appreciate the recommendation. Aside from the Blumenfeld book, it looks like it's the Human Brain book, the second book listed. So thank you very much. And it looks like we do have a comment in the chat box where another person had shared, and I'm not sure if you've experienced with this book, Dr. Lester, The Behavioral Neurology of White Matter by Christopher Filey. Looks like that book also discusses the behavioral aspects with regards to pathways. So Thank you, Mark. Appreciate that. Dr. Lester, is that a book that you're familiar with or one that you've referenced before?
No, I don't know that book, so I really appreciate the recommendation too.
Excellent. Well, thank you so much. And I think that concludes our time together today. Again, we really appreciate your lecture today, Dr. Lester. And we would like to let everyone know that next week, please join us for a lecture on networks, different brain networks, and that will be with Christina Vischer at 4:30 p.m. Eastern Standard Time. Thank you, everyone. Thank you, Dr. Lester.
Thank you.